Dear students, a very good morning to our attendances in USA and good evening to those joining us from Sri Lanka. We are delighted to welcome you to Ecoastronomy INC Astro Week Discussions and Awarding Ceremony 2025. Today marks a significant milestone, especially for our batch number 5 astrobiology and paleontology, as well as students from the astronomy and space science. On these special occasions, we are honored to introduce an esteemed guest speaker who will share a valuable insight during today's sessions. On behalf of our academic counselor, I will be the co-host on today's event. It's my pleasure to welcome our distinguished speaker. This is the time to introduce her. She's a paleontologist in training who will soon graduated from the Mount Holyoke University. Currently, she is served as a collection assistant at the Harvard Museums of Comparative Zoology and is a remote research member of the Viewer Lab at the University of Michigan. Furthermore, she is passionate about the mammalian paleontology and paleoenvironmental reconstructions. Today's our guest speaker focus on understanding how past climate changes event can provide a critical insights into our planet's future. Her fieldwork has spanned multiple geological periods and the locations, including Eocene in Wyoming, Cretaceous in Montana, Miocene in New Zealand, and Pleistocene in Canada, are reflecting her commitment to the uncovering a deep-time ecological dynamic with a strong foundations in both academic research and museum collections. She is poised to contribute meaningful to the fields of paleontology and climate science. Today, she will take us a journey into the tyrannosaurus, kiwi birds, and the series how to use vertebrate microfossils in paleo-environmental reconstructions. With that being said, I would like to invite one of the young aspiring paleontologists, Piper Hattenbach, to address Ecoastronomy INC International platform. Dear Piper, now it's your time. You can get started.
Hey everybody, I'm Piper. Thank you so much for that lovely intro, and I'm really excited to be talking with you guys today about, dinosaurs and paleontology and how all of those things kind of intersect. So hopefully you guys can see my screen now. Well, thank you all again for being here today, and yeah, let's get into it and talk about tyrannosaurs and kiwi birds and sieves and using vertebrate microfossils for paleoenvironment. But before I dive into that a little bit, despite that great intro, I figured you might want to know a little bit more about me. My name is Piper. Here's a bunch of pictures of me, looking nerdy about paleontology. I've been studying paleontology for a couple of years now, and I've been really grateful to get to do it all over. In that one picture on the lower left, you can see me with a bunch of Triceratops bones, or at conferences, or holding little lizard friends from the field. And my affiliations right now, was already announced, I work at the Museum of Comparative Zoology at Harvard. I'm on the Student Advisory Council for the Geological Society of America. Right now, I'm finishing up my undergraduate thesis in Paleocene mammalian evolution from the Weaver Lab at the University of Michigan, which is a really cool place that I feel really, really lucky to work. And in a couple of weeks, I should be graduating from Mount Holyoke College. So thank you to all of these places for, bringing me to where I am and for letting me talk today. And with that, let's dive into it. So paleoenvironmental reconstruction is kind of a giant word with a lot of letters, and it can mean a lot of different things. But I think just to start, I'm going to ask if you guys close your eyes for a second and you try and picture what you think maybe the Jurassic looked like, or what it looked like where in a world where dinosaurs lived. And maybe you're thinking about something kind of like this. This is a really great piece of paleo art made by Maxwell from Max Brontosaurus. And it's just a really beautiful way to maybe get to envision what life looked like about 60 million years ago. And maybe you also pictured something about this scene from Jurassic Park. And whichever way you were going, both of those pieces are the result of something called paleoenvironmental reconstruction. And paleoenvironmental reconstruction is really, really cool process in which basically paleontologists kind of act like detectives. We get to look at a lot of clues of what a past environment looked like and used a lot of different types of science to figure out what was there and why it matters. You need to know about botany to be able to figure out, like in that scene from Jurassic Park, what trees were there, what things grew here. You need to know about invertebrate fossils like shells and mollusks to be able to figure out whether there was fresh water or salt water, who lived in that water, and if that matters, why it matters. You need to know a lot about sedimentology, which is the science of the kind of layers that form in the earth from sediment deposited over time. If you're like me, you probably want to know about vertebrate paleontology and vertebrate evolution and how animals of all different types kind of came to be and came to grow. And you also need to have a pretty decent understanding of microfossils, which are usually these teeny tiny fossils and remnants from historic life, whether that's through diatoms, which are usually indicative of types of water, or isotopes, or pollen grains. And in paleontology, there are kind of two broad classifications of microfossils. There's the really, really small things. There's like diatoms or foraminifera, or little itty bitty shells in the ocean. And then there's a different classification of microfossils, which I like to call microfossils in quotation marks, which are usually small fossils that come from vertebrate animals. And for the purpose of today's talk, we're going to be talking about the other kind of microfossils. We're going to be talking about vertebrate microfossils, which are small fossils dropped by vertebrate animals. These really small-sized fossils usually come from vertebrate fauna, and they can be all types of things, like teeth, or scales, or scutes, or the little itty-bitty bones that exist between fingers and toes. But one of the really great things about vertebrate microfossils is that they can be usually distinctly identifiable. So even if you just find one little bitty thing about that big, you can take one look at it and know exactly who it belonged to and exactly who used to live there. And vertebrate microfossils often tend to form where they get dropped, often in a place of where there's a lot of water or a lot of moving sediment, and they can tend to kind of get smushed and pile up all on top of each other. So you can find these sections even within these massive formations where you can have hundreds and hundreds and hundreds and hundreds of these little vertebrate microfossils in one place. And using those hundreds and hundreds and hundreds of fossils, you can usually look at them all together and figure out a way to tell a story about paleo environments and what used to be there. And through those little itty bitty fossils, you can kind of, in your head, start to picture places that look just like this. You can see velociraptors and lizards and turtles and trees and lakes and all of these things layered together to make an actual ecosystem. And today I'm going to be talking about two different projects that we did where we used vertebrate microfossils to help reconstruct a paleo environment. We're going to be talking about one about dinosaurs and velociraptors and everybody's favorites. And then we're going to be talking about Miocene New Zealand. So New Zealand about 30 million years ago. And hopefully you find both of them interesting. And to get us started, we're first going to take a trip to the Hell Creek Formation. So for those of you guys in there who might be dinosaur nerds, you've almost definitely heard about the Hell Creek Formation before. It's the most famous formation in the world. It documents life in the Cretaceous, specifically in Cretaceous in North America, right along the coastline of the Western Interior Seaway, which is this ancient ocean that used to span right across the middle of the northern continent. And Hell Creek is especially famous because it's where the first ever Tyrannosaurus was discovered. It's where the first ever Triceratops was discovered. It's the formation that is most known for the biggest, craziest dinosaurs. And it also documents the moments right before all of those dinosaurs went extinct, and then life after, which is really cool. And just to kind of demonstrate where we are, we're in that little itty bitty corner of Montana all the way up there. I spent about 12 weeks out there doing this research. It was a really great time. And this was all on behalf of the Bureau of Land Management, which is a land management organization in the United States that manages public lands. A lot of the Hell Creek Formation is on lands owned by the Bureau of Land Management, so they help to fund and make accessible a lot of scientific research happening out there. And during the Cretaceous, things looked pretty different. If you look over at North America, you can see that giant Western Interior Seaway I was talking about. The Rocky Mountains are starting to form. Life was pretty incomprehensibly different. So the idea that we could go in there and just look at a bunch of itty bitty teeny tiny fossils and walk away with a better idea of what life looked like is pretty cool and significant. And specifically, these are the types of microfossils we're going to be talking about. All of these have, for this presentation, have been blown up to look a lot bigger than they are. But on average, almost none of these are going to be much bigger than a fingernail, other than a couple of the Tyrannosaurus teeth or the Triceratops teeth. And all of these little itty bitty fossils come from a wide variety of different specimens. If you look up in the upper right corner, you can see a really distinctive, shiny, kind of diamond-shaped fossil. That's called a gar scale. So that used to just be one single scale on a fish. Right next to it, there's a bunch of different types of fish teeth, little itty bitty fish vertebra, little shells and mollusks. So that's the invertebrate paleontology coming back in. Those kind of funky triangular looking teeth are all from different types of dinosaurs. We have early mammals, lizards. All of these microfossils can document a really, really wide variety of creatures. And all of these are from about 66 million years ago. And using that guide, we went out and we looked at a couple of different sites within this region called the Sand Arroyo Badlands to see how many different microfossils we could find and what they might be able to tell us. And because this was happening on public lands, we didn't want to collect any of these fossils. We wanted them to stay there where they, belong when you're not collecting with a permit. So we used this site called ArcGIS, which is a really, really useful piece of software to want to have if you're going to be a paleontologist. And using what we learned from ArcGIS, we went out into the field for a couple of days and mapped the whole area. After that, we picked 3 specific locations from the area that had really good microsites. Often these microsites were because they were in what's called a fluvial plain, which is pretty much a big fancy word for when you have a really, really large area that has a lot of water moving through it, but not always so regularly. So sometimes you have big crazy floods and then it's dry and dry and dry and dry for many months, and then a big crazy flood and then lots of drought again. But that can be really, really good for the transportation of small fossils because those big floods move all of those little fossils into one place. Often they kind of get caught in a nook or cranny. Sometimes if you're in a riverbed, maybe there's a big log in the way, and then all of these little fossils get caught right underneath the log and then covered in sediment and then turned into fossils. So we found a couple of sites that met those descriptions and we picked them. One had been previously identified by a different paleontologist who had come before, and the other 2 were new. And then over the course of so many weeks, we went out and combed through these locations and counted as many surface level microfossils as we could, and then counted them pretty much. Every single microsite was documented, quantified, and compared. And with all that said, let's get into it. So this is just a picture of my hand holding a couple of these little fossils, but hopefully this helps to just show you kind of the diversity we're talking about here. At the very bottom, we have a little piece of turtle shell, and right next to it, a crocodile scoot. Have you ever seen those bumps on a crocodile's back? That is exactly what it is. Today's modern crocodiles have these too. They're these little bony pieces of, we call them decorative osteoderms, which basically just mean extra fossils that give them those really, really distinct ridge shapes. There we have, right next to it, a really, really long-rooted mammalian tooth. An itty bitty salamander dentary plate. Those 4 little tiny fish vertebra were one of hundreds and hundreds and hundreds. There was such an abundance of fish vertebra. Next to it, we have another mammalian incisor. And then right next to that was one of my favorite things we found. It is this tiny, pinky nail-sized juvenile dromosaurid claw. And dromosaurid is the word for the family that Velociraptor and all of the other raptor dinosaurs come from. Because the fossil was damaged, we couldn't figure out the exact species, but we know that that is the itty-bitty claw of a juvenile dinosaur, and that's pretty radically cool. Next to that, we had crocodile teeth, guitar shark, guitarfish teeth, and gar scales. And that's just kind of a little snapshot into the diversity of fossils we can find here. And then we can actually further dive into what that diversity means. So the biggest thing, the most abundant thing we found was gars and other fish life. And that makes sense. Back in the Cretaceous, it seemed like this area was probably had a lot of lakes, a lot of creeks, a lot of moving water, and a lot of aquatic life. Gar scales, which are those kind of diamond-shaped things over to the side, are a really, really distinct fossil. They're really easy to spot. They're really shiny. and they preserve exceptionally well. And if you think about it, fish like gar have hundreds of scales. So one gar can result in hundreds of fossilized gar scales. Similar with fish vertebrae, they had the same problem. Fish have so many vertebrae. So one fish dying can really, really add to a fossil record in a really exceptional way. We also found some guitarfish teeth, which are pretty cool. Guitar sharks are still around today. Their teeth still look about the same, and it's really neat to see how little evolution has changed them. And about 48% of what we found belonged to gar and other identified fish. And that 48%, it tells us two things. Number one, it tells us, yeah, this was an area where there's a lot of water and there's a lot of fish living here. But also, we expected to find the most things from fish because that's what fish do. Like we were just talking about, hundreds and hundreds of gar scales and vertebra can emerge from a single fish. So it makes sense that they would have the most fossils represented here. Sorry that my slides seem to be lagging a little today. But this brings us to our next category, which is turtles and crocodilians. The next 48% of our fossils were from turtles and crocodiles. Kind of similar to like fish, turtles and crocodiles produce a lot of bones. Crocodiles are constantly shedding their teeth, and they have a lot of them. They also have dozens of scutes on their body, and turtle shells can break into hundreds of pieces. So in an area that was probably already more dominated by things like crocodiles and turtles, it makes sense that the fossil record would reflect that too. It's really easy to make a lot of bones emerge from those fauna. Doesn't make it any less cool though. But hey, it means I get to talk about dinosaurs again, so I'm pretty happy. And for this project, when we finally got to the dinosaurs, we only found 3 types of dinosaurs. And I actually think that makes a lot of sense in terms of the ecosystem, but I'll talk about that in a minute. We found 2 Tyrannosaurus rex teeth, which are pretty cool. These guys were relatively small and they were pretty damaged, but that also makes a lot of sense because if you remember, we weren't digging for any of these fossils. We were only looking at the surface for what is there. And when fossils are left on the surface for a long time, they tend to erode and change, and that definitely happens to these teeth. We also found Triceratops teeth. We found juvenile and mature adult Triceratops teeth, which was pretty cool. And microsite A, so one of the places we looked at, was located directly underneath a full skeleton of a Triceratops, which is pretty crazy. And you might be thinking right now, Piper, why didn't you guys excavate the Triceratops? And there's 2 reasons for that. Number 1, it wasn't in the best condition since it was on the surface. It had eroded quite a bit. And number 2, Triceratops are so abundant throughout this formation. You can find them absolutely everywhere. It's a little bit crazy. At the start of my summer out there, I was like, oh, I hope to just find one. That would be so crazy. And I think by the end of the summer, we had found maybe 50. It is incredible how many Triceratops skeletons there are out there. But that's a bit of a tangent. But then we found my favorite fossil for the whole site, which was this little itty bitty dromosaurid claw. Because it's missing its medial end, which is what you call the very, very end of the fossil, we couldn't identify it as a species, which was an absolute bummer. But finding things like this is pretty rare. We have a problem in the Hell Creek Formation that's called the small body preservation bias. Preservation bias is basically when there are issues or types of things that make it so that not everything gets preserved. And especially when you're talking about fossils, preservation bias is pretty huge. It's why we get so many gar scoops and so few of other things. But also when you have areas where sediment moves really fast, or where you have certain types of preservation that maybe favors things like plants, but doesn't favor things like bones, you can result in preservation bias, where some things get preserved and some things don't. So when you're looking at the fossils just from like a numerical perspective, it can be really tricky to tell the actual ratios of who was or wasn't there. And in the Hell Creek Formation, the type of preservation bias we have most prominently is called the small body preservation bias, in which small mammals and small dinosaurs and small animals really rarely get preserved in full. You might be able to find small pieces of them, like this claw, but because this is such an active area, meaning a lot of the sediment moved really, really fast, especially right after the asteroid impact, things that were much smaller than that kind of got crushed and didn't preserve so well. So being able to find these small body dinosaur pieces was really cool. But if we're going to talk about paleoenvironmental reconstruction, we have to talk about ecosystems. So just to like throw in the same statistics I've been saying the whole time, this is about the ratio of stuff we found. Everything in blue is somebody who lived in water. So that might be gar or other fish or guitar shark or turtles or crocodiles. And then the stuff in the warmer colors, like, oh, salamander should be in the water too. Sorry, guys. But then we found a smaller selection of things like dinosaurs and Tyrannosaurus and mammals. But when we actually think about how ecosystems work, this makes a lot of sense, especially with ecosystems that work now. Like, let's think about this in Sri Lanka. If you go to Yala, the thing that probably exists there the most is smaller things like birds and small mammals and squirrels. And then you build up from there, and then there's a lot of deer, and then you build up from there, and there's a lot of elephants, and then you build up from there, and there's only a small amount of like predators, things like leopards. There's a much smaller quantity of leopards than there are birds, right? Because that's how ecosystems work. You can't have, 100 leopards and 100 birds. The ecosystem would fall apart. So it was really neat to get to look at this and kind of see that same sort of ecosystem structuring 66 million years in the past with dinosaurs as we kind of have operating now. And because of this, we managed to kind of zoom out and be able to see a world that looked a lot like this. We could see these little feathered raptors, we could see T-Rex eating their food, we could see Triceratops and turtles and little lizards skidding around, we could see the lake that all of this stuff formed in, and the trees. And just for a second, even when all you're looking at are these itty bitty fossils, you can suddenly picture a whole ecosystem and a whole environment that you can't see when you're just looking at, the badlands and the dirt that exists today. And I think that's what's so neat about paleoenvironmental reconstruction, is that it lets you do that. And as promised, I have a whole second one to talk about, so don't worry, guys. Unfortunately, this one has no dinosaurs, so sorry, kiddos. But it is really cool for some other reasons. This is called the St. Bathans locality, and it's located in New Zealand. And I'll get into why it's so neat in a second. But first, a little spiel on New Zealand. New Zealand's a really cool place. It's famous for a lot of reasons. If you're somebody who's really enthusiastic about the outdoors, about geology like I am, you might first think about the crazy mountains and the beautiful hikes and the volcanoes or the adventure sports. Or maybe you're just like a really, really big fan of Lord of the Rings. But if you ask a biologist what they think is really cool about New Zealand, the answer you're almost definitely gonna get is the birds. New Zealand is really famous biologically for a couple of reasons, and one of them is all of its amazing, diverse, and crazy bird life. And you might be thinking, okay, New Zealand, that's cool, but we're in Sri Lanka. We have really cool birds too. But there's a bigger mystery here, which is that New Zealand has one total species of mammal that's endemic, meaning that it's from their island. Whereas Sri Lanka, another small island, you guys have hundreds of species of mammal. In fact, almost everybody neighboring New Zealand has lots of mammals. So why there's no mammals in New Zealand and kind of what evolutionary history brought us there has been a big mystery for a long time. And it's been a mystery that's been even harder to solve because New Zealand doesn't have a lot of terrestrial fossils, meaning fossils documenting what life on the land was like. Because New Zealand has two pretty big things going against it there. Number one, it's a volcanic island. And number two, it's an island that has gone underwater so many times. Often when there's been sea level rises, a lot, especially the South Island, has been ducked underwater. Especially pretty recently, about 32 million years ago in the Oligocene, there was this event called the Great Drowning, where almost the entire New Zealand island was underwater. So anything terrestrial that lived there before maybe didn't survive that. But luckily, there's been a solution to this mystery, to this terrestrial fossil absence, and that's called the St. Bathans locality. It is New Zealand's only terrestrial fossil record. It is the only place where we've ever found fossils in New Zealand that are older than the Pleistocene, meaning older than about 100,000 years ago, that tell us anything about what life on land was like in New Zealand. And that's really cool. So this locality shows us about a Miocene lake system from approximately 16 to 19 million years ago during what's called the Eltonian, but that's a fancy word you don't need to remember. Its climate was about 6 to 7 degrees warmer than today. So New Zealand right now, it gets snow sometimes, it gets pretty chilly in the winter, but back then it was tropical. It wasn't quite as warm as Sri Lanka, but it was getting there. And at the very, very end of the Miocene, so a couple, maybe 1 or 2 million years after everybody at the formation that we're looking at today, New Zealand cooled and it got colder, and a lot of the things in the a lot of things died off. The ecosystem changed pretty dramatically. So pretty much this formation is really cool because not only does it answer some of our questions about New Zealand's history, it's also a really temporary glimpse. It shows us this ecosystem that really didn't exist for very long, and that's a really rare and special thing to get to see. And with that, let's kind of dive into it. So for our project, we wanted to go to the formation. So if you look at that little red box on New Zealand, that's in a region called Otago, and that's where our site was. And there's actually hundreds of sites within this formation. This formation is really easily accessible right along the Metakorea River, which is a really long river running through Otago. And the river has pretty much eroded away the sediment and made it possible for us to actually get to access these fossils. So there's dozens and dozens of sites where you can go find these fossils right along that river. And we decided that we wanted to look 2 of them that were fairly far apart, but still close enough to be kind of in conjunction and related to each other to see what similarities or differences we could see about the fossils there. And we had to come up with some research questions for this because that's how science works. So our questions were, what are the quantitative differences in faunal diversity across the analyzed sites? Which is fancy science words for how many different types of fossils of different types of animals do we see at these places? Pretty much, does one site have more fish? Does one site have more birds? Why? Also, what are the qualitative differences in preservation of the documented fossilized remains? Which is, again, fancy science words for, does one site have fossils that were preserved better or differently than the other? Why do we think that happened? And what are the sedimentary differences between the sites? So pretty much, what does the rock that these fossils are in tell us about what this environment might have been like? And here's our methods. And you're going to notice that these methods are pretty similar to the methods that we used back in Montana at the Hell Creek Formation. This time we got to take the fossils out of the ground. But even then, the basic principles were the same. We went out and we sieved a bunch of sediment, which basically means we dug up a bunch of dirt and we put it in these big filter systems and in these big kind of like strainers, and we washed them through and we sieved it so that we only left with the bigger pieces and not actual just pieces of mud. And then we went through and we counted the bones and we separated the bones from what wasn't bones and we identified them. Identification took a really, really, really long time. So did sorting the bones. To do stuff like this, you have to be willing to sit at a microscope for hundreds of hours, which might sound pretty boring, but I think I'm exactly the right type of nerd for this. I had a really good time. I don't know if this sounds like anything you guys would enjoy, but I personally thought it was a blast to get to sit there and dig through these little bits of dirt and look for fossils. It was a blast. And after that, we pretty much went through and we just identified fossils. And the fossils that we were working with were small enough that we could fit hundreds of them into these little itty bitty petri dishes. So all of our fossils were sorted into certain quadrants of these petri dishes. They were labeled, and we absolutely took over the entire paleontology lab with these petri dishes. It took a lot. And from there, we actually got to learn some stuff. So let's get into our findings. Let's figure out what we learned. And you might have noticed those big circle things. Those are exactly the sieves that I'm talking about. They're essentially these pieces of wood with kind of like a type of net or wire stretched through it, and then you just wash off the dirt. So one of our sites was called HH0. This picture of that site, and HH0 was located pretty low and right along the creek. And there's a couple pretty cool things about HH0. One of them was these things called thrombolites. So if you see that red layer, fossil layer that we drew on, if you look right above it, you might see some funky looking rock. And that rock is called a thrombolite. And if you're like me, you've probably never heard the word thrombolite before, but maybe you have heard the word stromatolite. And that's because they're pretty much the same thing. Thrombolites and stromatolites are both rocks and living organisms that form from layers and layers and layers of these algae, pretty much. And it's one of the most ancient forms of life on Earth. Stromatolites usually form in oceans, whereas thrombolites are the freshwater variety, is their big distinction. And these guys, they're old, but they're not the most ancient form of life on Earth. These are thrombolites. These guys are only 16 million years old at this location. But what made this so cool was that our fossil layer was pretty much sandwiched between layers of thrombolites. And if you look at that little zoomed in picture of our sediment and where we're finding our fossils, you can see a couple of little itty bitty fish vertebrae, but almost all of that kind of white gray material, that's all thrombolite. All of those are little fragments of thrombolites, little fragments of that algal mass. And overall, we counted 2,226 fossils from this site. And it was pretty dominated by fish and by birds. 38.5% of what we found was bird, and 60% was fish. But if you look at the side, you can see some of our other cooler fossils. We found little bones from raptor birds, so things like eagles and hawks. We found eggshells from these ancient, now extinct birds called moa, which were pretty much these like sometimes 12-foot tall, like ancient prehistoric birds that kind of look like emus, but way bigger. And their eggshells are really distinct because they're pretty thick and because they have really unique pores. So we used to find a lot of these eggshells in the sediment. We also found Raptorian toe claws, so kind of just like that dromaeosauride claw from Montana, except this one is a toe bone from a modern raptor, or from a 19 billion year old raptor from what we think might have been a giant eagle. And then we found little pieces from crustaceans too. And if we go over to Crocsite, you'll notice a couple big differences right off the bat. For one, this isn't right along the river. This sediment was kind of elevated and pushed up into the air and separated from the rest of the river, which means that its sedimentology is going to be a little bit different. It was much more dominated by finer grained sandstone, and all of the thrombolite pieces that were there were pretty worn down and much smaller. And in that picture, you can see in our tweezers, we're holding up another piece of moa eggshell. At this site, we found 2,100 pieces of identifiable material. So when we say identifiable material, that just means fossils that were preserved well enough that we could actually figure out what they were. And this site was absolutely fish dominated, especially in comparison to the other site we just looked at. In comparison to HH0, you can see there's a huge difference. Almost 87% of what we found was fish. We found some other really cool stuff too. You know, we found a couple KIWI radii, which are these little itty bitty bones in kiwi. But for the most part, it was fish, fish, fish. So if we want to go into our comparisons of what species we found, specifically species of birds, which are some of the most distinct and cool things we could find, you'll notice a trend pretty quickly, which is that Crocsite, which is the one that had the most fish, also has the most water birds. We had the most ducks, we had the most rails, we had the most shorebirds. That site was really, really dominated by birds that live in or near the water, with the exception of the kiwi bone. That one's kind of throwing us off a little bit. Whereas HH0, by comparison, had a lot of bones belonging to birds that either live right along shorelines specifically, or much further on in the forest, you know, things like eagles and parrots. And that distinction is totally going to matter when we're thinking about what differences might be said about these two locations. And similarly, HH0 had way more fish. Crocsite had way more fish. I get them mixed up all of the time. And HH0 had way more birds. And when you look at this comparison, you can kind of almost immediately feel, so one site had a lot more water bird species and a lot more fish, and the other site had a lot of birds and a lot less of everything else. I wonder what that says. And it's not, exactly a genius thing to figure out. But what we realized is that one of our sites, HH0, must have been right along the shoreline, whereas our other site, Crocsite, must have been further out into the actual lake itself. But this was even further proved by those thrombolites. Remember those big algal round masses? It turns out that thrombolites only form right along shorelines and in really, really shallow water. So the presence of the full thrombolites themselves suggests that not only was HH0 near the shore, it was the shore. Whereas our other site, Crocsite, might have been further out in the lake itself. And from an ecology perspective, that's really cool. One thing about studying lakes is that lakes are separated into a lot of different niches. The stuff you're going to find right at the shore, or even a little further out at the shore, or even 5 feet deep versus 10 feet deep versus 15 feet deep, are going to be totally different. And it's really crazy that we could see that niche and that level of change from 19 million years ago, just from looking at some itty-bitty fossils. So anyways, this isn't exactly our most beautiful figure, but I think it demonstrates that really well. And once again, we kind of have that moment where just by looking at a small handful of these little itty-bitty fossils, all of a sudden we can see a world that looks like this. We can see these extinct species of hawk diving down. We can see hundreds of different species of duck crowding these floodplains and these lakes. We can see seagulls, we can see parrots, we can see moas eating their lizards. All of a sudden, a really different world is possible. And you might notice a crocodile in the background there. Even though we didn't find any actual crocodile material at the site, there was crocodiles here before. Other people have found them, which is really significant because New Zealand doesn't have crocodiles anymore. They're too cold for crocodiles. So this really was a really, really different world. And that's really cool. Additionally, before we did the study, there's a lot of research still ongoing and will be ongoing for a long time at this site. But there's always been a lot of debate about whether this site documents a lake or a floodplain. Basically meaning, is the water there all the time and is that a supported ecosystem? Or does it just get wet sometimes and then all the water leaves, then it gets really wet sometimes and all the water leaves? And this study really, really helped us to add quite a bit of evidence for the lake side of things, to the fact that there are these long-term aquatic ecosystems existing here. And then that shows up in the distinction and disparity between what species are and aren't present. So let's talk about similarities between these projects, because there's definitely quite a few. Both of these were short-term projects that took 6 months or less. So even if you don't have a lot of time, you can still you can get science done. You can do quite a bit. The vast majority of the time was spent on specimen identification and on figuring out what fossils we did or didn't have. They're both collaborative and interdisciplinary. So one of my favorite things about paleontology is that it's a science that's interdisciplinary, meaning it's not just one thing. And you have to be willing to connect with a lot of different scientists from a lot of different backgrounds. That St. Athens project right now, there's now, let's see, the 2 people leading it are both pretty much bird experts. That's what they study, but they're now bringing in sedimentologists and chemists and traditional paleontologists and all sorts of people from all sorts of backgrounds and botanists to help answer questions. And that's pretty much a lot of what paleoenvironmental reconstructions are about. You have to know things about modern day ecology and modern day biology, and also historic botany, and also sedimentology, and also paleontology. And all of these pieces that kind of all fit together really, really nicely. It lets you experiment with a lot of different types of science and dip your toes into a lot of different types of science and kind of learn a lot from the people around you and the people you're working with. They also have really basic methods for the most part. Almost entirely the science here was, let's find some fossils and then let's count the fossils. And that's one of the really cool things about microfossil science, is that it's pretty easy to get involved in. You really just need to be willing to spend a lot of time on it. And for both of them, you had to think about preservation bias. So like we talked about earlier, it's never quite as simple as, oh, we have, 50 crocodile teeth. That means there's 50 crocodiles. Well, one crocodile could drop 50 teeth. Or those teeth could have come from 4 crocodiles. It's very difficult to be able to determine those things. So you have to think about preservation bias a lot when you're doing that kind of work, and think about why the fossils that you're looking at ended up where they are. But even if it sounds really simple, microfossil science and the examination of vertebrate microfossils is really important and really central and really, really widespread. This is a picture I took last year when I was attending a conference in California. And we stopped by the La Brea tar pits. I don't know if you've heard about the La Brea tar pits, but it's one of the coolest fossil sites out there. It's located in California. They're really famous, and they do really, really cool research about Pleistocene ecology. And if you see here, there is a penny, which is a little coin, tiny, tiny, tiny. And even tinier are these little fossils, zoomed in and magnified. And this is an active vertebrate microfossil sorting station that they have at the La Brea tar pits, going every single day, all of the time as they identify these little fossils and figure out what they are. And just on the screen, you can see such a diversity of life. You can see that little claw that looks a lot like our little raptor claw from before from a bird. You can see a lizard jaw. You can see seeds. You can see coprolites. You can see snail shells. You can see a little itty bitty humerus. And all of those pieces come together to tell us a much bigger story. So at a lot of really famous sites around the world, people are always hiring people who are willing to sit at a microscope and pick through and look for these little fossils. And those little fossils can contribute a lot to our science. So even if it seems kind of simple, this is a really easy and really great way to start your scientific journey and get connected and get started. And that's it. That's my talk. Thank you guys so much for having me. And if you have any questions at all, I would be more than happy to answer them.